Could you please give an enormous, warm Monday morning welcome to Professor Annette Haworth.
Thank you, Chris. Yes, that's a little bit different to the theme that we've had last week, and over the next hour I'm just going to throw some slides at you just to give you a bit of a feel for the things that we do as medical physicists, and then at the end of the hour you can shoot questions at me and maybe we can expand things in the direction that you'd like to go. So I've got here the title, Madame Curie, Schrödinger, and we're going to talk about the day in the life of a medical physicist. So what we're going to do in the next hour, I'll give you a little bit of an introduction of how I got into medical physics, and I'll talk a lot about radiotherapy physics because that's the area that I specialised in when I was working in a hospital. We'll go through the life and the day of a medical physicist, some of the things that we might do. I want to share with you some of my particular research interests, and we'll wrap up by telling you what I love about being a medical physicist. So I came from the UK, I did my undergraduate degree in Leeds in England, and then I moved to Australia and I started doing my clinical training in Perth at Charles Gardner Hospital, which is where I did my master's and my PhD as well. So in those days we used to do all of our training on the job. It takes about 5 years before you are competent enough to practice as a clinical physicist, and you have to sit exams and practical exams and so on and so forth. These days it still takes about 5 years to be a fully certified medical physicist in practice, but you have to now formally go through a master's program as well as clinical on-the-job training. Okay, so after spending 10 years in Perth, I moved to Melbourne. I moved to Melbourne and worked at the Peter MacCallum Cancer Centre, which is in Melbourne, and Peter MacCallum Cancer Centre is Australia's only public hospital solely dedicated to cancer treatment, research and education. So I completed my PhD in Perth and then moved to Peter Mac because they offered me a part clinical and part research job, which is exactly what I wanted. So I spent 10 years there, and then I moved to Sydney in September last year. So as Chris pointed out, I'm now the director of the Institute of Medical Physics, so I've sort of moved from a clinical position to an academic position. I've got probably one of the best offices in the world, sitting up there in the Ivory Tower. I've got a fantastic view and part of the beautiful School of Physics building. So in this job's quite an interesting job. I wear many hats, so of course I'm the director, and I'm also the course coordinator for the Master of Medical Physics program. So this is one of the few programs in Australia that is accredited, and having this master's degree plus clinical training will allow you to practice in hospitals as a medical physicist. Obviously I supervise PhD students, or research students, as you would expect. But my job's a little different to many of the other professors in the university. My job's actually part funded by the Ministry of Health. And in that capacity, my job is to interact with all hospitals that deliver radiotherapy services in New South Wales. So I advise the government on technical issues, and I work with hospitals to help them with research, development, and any other things that I can do with my background. Now, what does a medical physicist do? Now, it's really hard to explain because we actually do lots of things. There's actually lots of careers in medical physics. But if I tried to simplify it and just put it into 4 buckets, this is what I would come up with. So most of us, or probably a large proportion of us, work in radiation oncology, and that is treatment of cancer with radiation. In fact, that's the area that I've worked in my professional career. You could be involved in nuclear medicine, which is mostly to do with imaging with radioisotopes. We also have a field which is called theranostics, where we can also treat with radioactive isotopes. Radiology is where we're involved with mostly diagnostic imaging using X-rays, ultrasound and MRI. Or you could be involved in radiation safety, which is about making sure that members of the public and staff are protected from ionising and non-ionising radiation. We're also involved in making sure that the patients themselves don't receive any more radiation than is absolutely necessary if it's a diagnostic test, or they get exactly the right amount of radiation if it's a therapeutic administration. So as Chris said, I've got some gory slides in here. I hope you all enjoyed your breakfast this morning. Please hold on to it. I have selected the slides, so hopefully there's not too much, but sometimes when you've been working in the field for a long time, you just forget that people aren't used to this sort of stuff. But anyway, I'm sure you've all watched Embarrassing Bodies or something, so here's what we'll see. So I thought I would...
That's all right, all right.
So this is actually sort of a good way for me to start, because really I've just made the point already that there's these 4 categories of medical physics. Well, I actually started off doing electrophysiological testing, and this involves essentially putting electrodes on the patient's head or some part of the body. What we're doing is we're measuring the tiny electric signals that you get through the nervous system. So we were looking for interruptions in that nervous pathway, and that would help us diagnose what was going on. And I also did a bit of diagnostic X-ray work on the side. So this is doing quality assurance. QA stands for quality assurance. So what we're doing here is we're looking at how good the image quality is coming out of this X-ray unit. So what you can see here are some lines, and you can see that they are distinct lines. But as they get closer and closer together, suddenly they become a bit of a blob. And this is what we call image resolution, and we count the number of line pairs that we can see. So we would say, well, we can just resolve the line pairs on that image, so that's the image resolution for that particular setting of the diagnostic machine. So after doing that for a few years, I moved into radiotherapy. So I thought, how can I simply explain to you what radiotherapy is, and like everybody else, I hit Google and said, what's radiotherapy? And this was the definition that Google came up with. It says, radiotherapy is the treatment of disease, especially cancer, using X-rays or similar forms of radiation. That's actually a pretty good explanation of what radiotherapy is, so I thought we'd go with that today. Just a bit of terminology. Patients go to radiation oncology departments to receive their radiotherapy treatments. Well, I have to admit that we use the terms interchangeably quite often, but strictly speaking, those are the terms that we shouldn't be using. So where do these X-rays or similar forms of radiation come from? Well, these days most patients that are having radiation treatment for their cancer comes from a linear accelerator, or a LINAC for short. So I'll keep saying LINAC because that's the terminology that I'm used to using, but LINAC is short for linear accelerator. So LINACs produce high energy X-rays, and we'll have a look inside a LINAC in a moment. Radiation can also come from a brachytherapy source. So I've actually got a show and tell for you. So at the end of the lecture, if any of you are interested in having a look, this is not radioactive, obviously, but you can come and have a look. But essentially what we've got here is a radioactive source. It's only the size of a grain of rice, about 5 millimetres long on the end of a cable. And the radioactive source lives inside this housing, so you can see there's the radiation sign up there. And under computer control, we can send that radiation source, which is on the end of a long cable, down through this long tube, which is called a catheter, into the patient. And what it does is it steps through the tumour. So the catheter has gone into the tumour, and the radioactive source steps through the tumour, delivering the radiation as it goes. I thought I'd briefly mention protons to you because Australia is just about to have its first proton facility installed in Adelaide, but I won't spend too much time on protons. But essentially what you can see here, here's a patient lying on a couch, and the protons actually come from behind this area here. You might wonder what's actually behind this area here. It's this. That's pretty amazing, isn't it? That's called a synchrotron, and that's where we produce our protons or heavy ions. And what happens is the protons come out and they whiz around this track here at very high speed until they're ready to come out and get blasted at the patient. So a little bit of physics now. So how does radiation work? What's this actually doing? So you all know, of course, within the body we have DNA. So this is a normal DNA string. If we have X-rays, the DNA can come in, so it's a double helix, and so we can either have what we call single-strand breaks, where we just break part of the chain, or we can have double-strand breaks, which happen not quite so often, and that would completely destroy the cancer if we had a double-strand break. With proton beams, they hit pretty hard, and most of the time they will just knock the DNA out. So in other words, that kills the cancer. And if we use heavy ions, which can be carbon ions, then they really do give quite
quite a punch. Again, a little bit more physics. So how does it work? The principle behind it is Compton scattering. I presume you haven't covered that in your high school physics yet, but you will possibly come across it. So depending on what energy of radiation you're using, this is the area of the energy that we work in radiotherapy physics, where Compton scattering is the predominant physical property. So essentially what happens is X-rays come in from our LINAC. They strike electrons within our body. They produce scattered electrons, which can then go on and perform further ionisation, or it can produce a scattered radiation, which again goes on and creates a cascading effect to deliver the radiation and harm the DNA in the body, or the tumour, hopefully the tumour. So this is a schematic of what a LINAC looks like then. So here's a patient lying on a couch. This is called a waveguide. Electrons accelerate down a waveguide. The electrons, of course, have got a charge. So in a magnetic field, we can rotate them through 90 degrees. We send them through, they strike a target. So the electrons strike a target to produce the X-rays, and they then pass through a collimator, and that shapes the field. So it matches the shape of the tumour. And so the tumour, of course, is sitting deep inside the patient. What we do is we actually shoot the radiation from lots of different areas. So we're shooting the radiation beams, so they're focusing on the target. So the thing to be aware of course, is the radiation has to pass through normal tissue and to hit the target. So by bringing it in lots of different directions, the healthy tissue around the tumour gets a little bit of radiation, but most of the radiation is focused on the tumour. So what we're going to do now is have a look at a little video to show you how a LINAC works.
How the linear accelerator works. All body cells can be damaged or killed by radiation, but tumor cells are more sensitive to radiation than normal cells. Radiation therapy uses this principle to damage beyond repair or kill the abnormal cancer cells in a tumor. Successful radiation therapy depends on the ability of the linear accelerator, or LINAC, to deliver a tumoricidal dose of radiation to the tumor, while ensuring minimal radiation of normal tissue. The LINAC accurately produces, monitors, controls, and conforms the radiation beam to the planned target. Beam generation. Radio frequency waves are pulsed into the waveguide by the magnetron. This is synchronized with the injection of electrons into the waveguide by the electron gun. The radio frequency waves accelerate the electrons along the waveguide to a speed approaching the speed of light. The X-ray beam is created when the electrons hit and interact with a tungsten target at the opposite end. The magnetron controls the power and frequency of the radio frequency waves, which determine the energy of the X-rays produced. The digital accelerator uses a diode-type electron gun situated at the end of the waveguide. The electrons are produced by heating the tungsten filament within the cathode and are then injected into the waveguide. The number of electrons injected is controlled by the temperature of the filament.
Okay, so that's radiotherapy using LINAC. So what we'll do now is have a look at brachytherapy. So this brings in Madame Curie, so that was in my title. So Madame Curie, she won the Nobel Prize for discovering radium and polonium. Radium was actually the first radioactive isotope that was ever used for radiotherapy treatments, and so it goes back to the days of Madame Curie. We don't use radium anymore. It's not a very nice isotope to use. It's quite toxic and very difficult to handle. So that's been replaced by more modern isotopes that we produce in, typically we produce in nuclear reactors, which of course weren't around in the days of Madame Curie. So the idea behind a brachytherapy is we place the radioactive source in or near the tumour. This is a radioactive I-125 source. Again, it's about the size of a grain of rice, and we can permanently put these inside tumours, or we can temporarily put them over the tumour surface if that's what we want to do. So the important thing to know with brachytherapy is that high radiation doses can be delivered to the tumour while sparing the surrounding healthy tissues. So with radioactive sources, the radiation drops away very, very rapidly as we move away from the radioactive source. So if we do a head-to-head comparison of brachytherapy and external beam radiotherapy, so this is all of those sources. So we've got about 100 sources here. Now they're actually implanted in a prostate. Prostates are only found in men, and we'll go into the anatomy of that in a moment. But essentially they're in the pelvis. This is the pelvis here, and here are the seeds that have been implanted into a prostate that's got prostate cancer. So the radiation comes out from the sources, but it doesn't go very far. It gets absorbed very rapidly by the actual prostate tissue. And that's in contrast to external beam radiotherapy, where, as I said, the radiation comes out of the head and passes through the body before striking the target, which is the tumour, and then exiting the body as well. So what does it look like? So this is actually a dose distribution. So just to explain the differences in terms of how much dose is deposited in a patient when we're actually delivering radiotherapy. So on the top left here, this is what is for external beam radiotherapy. It's a special technique called VMAP, but essentially it just means that the radiation comes in from lots of different angles. We're trying to hit the target in the middle here. We're trying to have the highest dose here, but the blue indicates a low dose bath. So there's radiation that is being delivered to the whole pelvis here. It's a low amount of radiation, but it's still there. And of course, any amount of radiation that you deliver to the patient, it comes with it a risk of secondary cancers and damage to healthy tissue. This is what a proton or a heavy ion treatment looks like. So again, the radiation is focused on the tumour in the middle, but it just now passes through two different angles. So you still get some radiation, low-dose radiation passing through. But if you compare this with brachytherapy, so this is low-dose rate, where we put those radioactive seeds permanently in the prostate, and you can see we've got a very high red spot there, so lots of radiation and not much of that low dose bath here. And this is high dose rate, which is where we use that source on the end of a cable and the robot to deliver the radiation. Again, a huge red spot right in the middle, exactly where you want it, with not much dose spilling outside the volume. So here's the gory picture. So this is one way of using brachytherapy. We can use for many different cancers. This is for breast cancer. So this patient has almost certainly had surgery. They've had the tumour removed. But what can happen is we can leave cancer cells behind. And so what we like to do is deliver a little bit of radiation to the tumour bed just to make sure that we've mopped up any remaining cancer cells. So what's happened here is we've got a whole pile of catheters through the breast and into the tumour bed. So as I said before, the radioactive source comes out and it travels down the catheter, it steps its way through the tumour volume, it goes back into the housing, and then it goes down the next catheter and again steps through the tumour volume until the treatment is complete. So that's what the radioactive source looks like on the end of the cable. In terms of the seeds which we put permanently into patients, so this is an example of using it for prostate cancer, what we do is we put a ultrasound transducer in the rectum, which is just here. This is the rectum. Here's the prostate, so it just sits above the rectum. This is the bladder here. And we put needles, so the patient's asleep, of course. We push the needles through, unless they don't behave themselves, and then we keep them awake. We push the needles through, and then we drop all the seeds into the prostate, and then this is what it looks like at the end of the day, once all the seeds are in there. Now, the patient is sort of radioactive, but they're not green. But actually the radiation that we give off is very low energy, so all the energy from the radioactive sources is concentrated in the prostate. So if I got a Geiger counter, I could go around the patient and I could measure something, but it's not a lot. But we keep the patients away from children for a few months, at a distance of 1 metre or more, but really they're safe to go out into the community. Now, the exciting thing about this, and this is why I harp on a lot about this, is because I was the first physicist to do this in Australia, and it had been done in a few centres overseas, but so we decided that we would do it when I was working in Perth. So In those days, I had to do everything using graph paper and a calculator, and we didn't really know what we were doing, but we just knew we had to get the radioactive sources into the prostate. And so over time, I was asking lots of questions. What's the best way of doing this to make sure it's effective and what low side effects? And that resulted me in getting a PhD for this pioneering work. And we still do this today. We started doing it in 1994, so we've been doing it for many years now. This is actually how we used to do it before we used the ultrasound technique. Now, I don't know whether this is a true story, but this is the story that I've been told. But what we used to do to put the radioactive sources into the prostate was the registrar, who's the junior doctor, would have to put his finger into the rectum, and then the surgeon, the esteemed surgeon, would push the needle through into the prostate until the registrar said, ouch, and then we knew the needle had gone far enough, and then he could drop the seeds out into the prostate. But that didn't really work very well because you'd end up with some bits of the prostate not covered with radioactive sources, so that wasn't a very good treatment. So I've taught you now about external beam radiotherapy, I've taught you about brachytherapy. So what do? So we're actually not very good at telling people what we do. So I've tried to sum up in a few sentences what we do. So in terms of a radiotherapy physicist, we make sure that the right amount of radiation gets delivered to the right place. Okay, so we want to make sure that the patient gets the right amount of radiation and we deliver it to the tumour and we don't miss. Okay, we don't want to put too much in there because we might destroy cells that we don't want to. We don't want to give too little, else we may not cure the cancer. We also spend a lot of our time working on research and developmental projects, so we're continuously looking for ways of increasing tumour control and decreasing the side effects from treatment. We also make sure that other people don't get irradiated as well. So we have to make sure, for example, where we put a linac, we put them in a room which is called a bunker, and these bunkers have really thick walls. They're like a metre thick. That's how much radiation comes out of these machines. So we do the calculations to make sure that the staff and the members of the public are protected as well. So all physicists or clinical physicists at some point will be involved in quality assurance tests, and it's true that we do spend a lot of our time associated with quality assurance issues. So here's a physicist working on a linear accelerator. We physicists like to think all patients look like square blocks, and these blocks are water equivalent because we think patients are made up of water. So we take a very simplistic view of things, and what this physicist is doing now is measuring the dose of the amount of radiation that's coming out so that we can make sure that patients get the right amount of radiation. Now, the thing to understand is that these machines, this is the gantry here, the linac, rotates around a point in space. Even though it weighs several tonnes, it actually rotates about a point in space, which we call the isocenter. Now, this point in space is about the size of a pea. In actual fact, it's even smaller than a pea. We have to make sure that when it goes around, this point in space has a diameter of less than 2 millimetres. And in fact, this machine, this is a super special machine, it rotates about a point in space which has a diameter less than 1 millimetre, so it's super high precision. And that's the sort of precision that we work to in radiotherapy. We work to 1 or 2 millimetre precision. So how do we check that this huge machine is focusing the radiation at a point in space which is no bigger than 1 millimetre? That's actually a very difficult thing to do. So this is a test that we do. So we have here a ball bearing which is placed at that isocenter, at that point in space that the gantry rotates. So the radiation is coming out of the head. The ball bearing is here, and we have some film here. So when the radiation comes out, this is what you see on the film. So this is the radiation that's coming out of the head, which is a square block, and this is the shadow of the ball bearing. Now, if the ball bearing is actually at the isocenter, it will be in the center of that square. And you can see it's a little bit displaced, but not very much, less than a millimetre. If we move the gantry around to another angle, we shoot another film, and so we can see this is what we call the isocenter run out, and that can tell us how big that sphere is that gantry is rotating in space. And in this case, it's better than 1 millimetre in all cases. Now, in terms of developmental projects, I've been involved in many developmental projects, and obviously I couldn't cover everything, so I'm just going to give you a little bit of an example of a project that I was working on in Melbourne just before I moved to Sydney. This is a treatment involved with treating cancers at the back of the eye. So this is an MRI image. Those are the two eyes, of course. Now, this particular patient has a tiny tumour right at the back of the eye. It's less than 1 centimetre. In fact, it's 6 millimetres in diameter, and we need to treat that tumour. In fact, the problem here is that this is a different patient, obviously, because it's the other eye, but this tumour here is outlined in yellow. We've got the optic nerve leading from the eye. Now, if we put too much radiation on the optic nerve, the patient won't be able to see. This is the lens at the front of the eye. If we put the radiation through the lens of the eye, they'll get cataracts. They won't be able to see. We also have a thing called a lacrimal gland, and the lacrimal glands produce our tears. And so if we irradiate the lacrimal gland to too much radiation, then the patient won't be able to produce tears there, and their eyes will dry out. So it's a very challenging tumour to treat. But also, when we put the patient on the linac, we can immobilise the head, and in fact, this is what we use. So again, you can come down and have a look. You'll see this in a slide in a moment. We can keep the head still, but how do we stop the eye from moving? So what we were doing was developing a system that uses a periscope. So here's the eye here, and we get them to look straight up. This is a mirror, and they see a red dot. The red dot actually comes from an LED, which is back at the end of the periscope. And so we ask them to fixate on that red dot. And we have a camera here, which means that we can watch them. So if they start drifting off, not looking at that red dot, we know, and we can stop the radiation beam. So this is what our prototype looked like. So here's a patient wearing this mask that you can see down here. This is the frame that we built. There's the periscope back there, and the patient is looking at the red dot. These balls here are actually infrared reflection balls, and we use those to set the patient up on the linear accelerator. This is the final version of the system that we developed just before I left. So here's the camera. We actually did away with the periscope, but the mirror system is here, and then we could display in the room on a laptop that the patient was set up correctly. And this is sort of a schematic view. Well, it's not a schematic, but it's an early view of what we were doing. So we can see a grid system here. So when we did all of the preparation for the treatment, the patient's eye was centred around a circle here. But you can see the eye is a little bit off. And so what we have to do is get the patient to bring their eye back into the right position before we treat, so that we know that that tumour is positioned at the isocentre, so that we can then focus the radiation beam right onto the tumour. And again, we're looking for a precision better than 1 millimetre. So I've been involved in a number of other projects over the years that I thought I'd just mention. This one is actually for treating leukemia, and I got my MSc out of this particular project. Now, the point is that with radiotherapy, normally we treat tumours, solid tumours somewhere in the body, and we focus the radiation fields on that. If the patient has got cancer all through their body, they've got metastases, then normally they would have chemotherapy. In the case of leukemia, that's a cancer of the blood system, so normally the patient would receive chemotherapy. In fact, they always receive chemotherapy. But sometimes the chemotherapy doesn't quite work. And so what we have to do is we have to give the patients a bone marrow transplant. That's where the blood cells are produced. And what we do is we give the patients a very high dose of radiation to the whole body. In fact, it's a lethal dose of radiation. If we didn't give the patients a bone marrow transplant after irradiating them, they would die. So can you imagine how it felt the very first day that we pushed the on button, and it was a child, because we used to treat children, and giving them a lethal dose of radiation. Can you imagine how that felt? Anyway, as far as I know, no patient died as a result of the work that I did. The other thing I thought I might mention, this is more recently, is I was working on a thing called SABRE, which stands for stereotactic ablative radiotherapy. Ablative means we give super high doses of radiation very, very quickly. And this made it on the news. We were on every channel on the news that night that we did the first treatment. So I'm going to show you a video now, and you'll see that's me there walking in with the radiation oncologist for the very first treatment, and the patient, of course, is leading the way in the blue gown. So I'm just watching on because that's what we've done. I've done all the hard work leading up to this treatment, and now I'm just there to make sure everything is going to plan and answer any questions as we go. Oops.
80-year-old Harold Severus is the first patient in Australia to undergo a new cancer treatment, but he's not nervous because it will be over in record time.
I feel pretty good. Excellent.
The ability to be able to deliver this type of treatment in a single visit in under 15 minutes, it's super convenient for the patient.
Harold is about to receive a single high dose of radiation targeting a tumour in his lungs. Conventional radiation treatments are delivered daily over a 6 to 8 week period. Now that's been slashed to a single 10-minute radiation dose using this new procedure known as tailor therapy. He lays wide awake and perfectly still alone in the room while outside the medical team at the Peter MacCallum Cancer Centre manoeuvre the machine. They locate the tumour, then carefully zap the cancer with a beam of radiation. This is an extremely precise treatment with less than 1 millimetre pinpoint accuracy, and it's very effective with over 90% success rate. Soon it's all over.
For him to be able to come in for a single treatment and go home the same day and not have to worry about anything after that, it's really great.
It doesn't feel as if I've had anything done at all. So, yeah, how good is that?
For now, the rapid treatment is only available for certain hard-to-treat cancers such as lung and liver. This grateful patient is still stunned at how fast the gifted medical team and their machines now operate.
Just sit back and relax. That's all there is to it.
Emily Rice, 9 News.
Okay, so that sort of gives you a bit of a snapshot of what a treatment actually looks like. Of course, that was delivered in rapid time, which was the whole point of the little news clip there. So I thought before we go any further, you should perhaps have some feeling for what a patient goes through when they've been referred for radiotherapy. So a cancer patient would almost certainly have been to see a GP at some point. They've got a problem, and the GP has sent them off for some tests and worked out that the patient has cancer. And so at that point, the GP will refer them to a specialist. The specialists that they may refer them to include a surgeon, who of course chops it out, a radiation oncologist who wants to zap it, and a medical oncologist that wants to give chemotherapy. And there are other professions as well. Sometimes the patient will get to see all three, or maybe just two of the three groups of specialists. And then what happens is the specialists all get together at a meeting which is called a multidisciplinary team meeting, or an MDT, and the physicists go along to that, and we work out what we think would be the best treatment options for that patient. The doctor then discusses it with the patient, and the patient has the final say on what treatment they want to have. So assuming they're going to go for radiotherapy, they will then be under the care of the radiation oncologist, who is the doctor. The radiation therapists actually administer the treatment. They also do the calculations for the treatment. And then the radiation oncology medical physicist is involved at various steps along the way, which is what I'm covering in this talk today. So the first step for radiotherapy planning is for them, usually they have some sort of imaging, and it's usually a CT scan these days. So a CT scan is X-rays, and it gives us a three-dimensional view inside the body. So what we can do is we can see where the target is. So in this case, again, a prostate, which has been outlined in red. The bladder is outlined in yellow, and the rectum is there in that blue colour there. And so the bladder and the rectum are radiosensitive organs, and the prostate is what we want to treat. So what the radiation therapists will do is they will do a plan which brings in the radiation beams and minimising the radiation dose to the rectum and the bladder, but focusing all the radiation onto the prostate. I'm going to skip over this one, and then eventually, once the treatment plan is done, the patient then goes to treatment. So I'm skipping over that one because I've got this slide which explains the process here. So what happens is, before we treat the patient, we do some imaging. And what this is showing is, this is a CT scan of the patient's head in this case, not a prostate of course. This is the patient's CT scan, and this is what we used to do the treatment plan, all the calculations for the treatment. And the bit in blue is where the patient is on the couch today relative to where they were on the CT scan. And you can see there's a bit of misalignment in the images there. So we shift the patient until it all completely lines up, and then we know that the radiation is focused on the tumour. So this is what a treatment plan looks like. So we had a look at one before when we were looking at protons and brachytherapy. So essentially we're making sure that the patient is going to get the amount of radiation that the doctor has prescribed, and we make sure that the normal tissue is not getting too much radiation dose. I'm not quite sure what part of the body this is supposed to be treating. It's probably a cancer of the cervix, but essentially it just gives you an idea of what we're actually looking for. But the point of this slide really is to say, well, we've got a computer that does all these fancy calculations, but you've got to give the computer data. It needs to know how much radiation is coming out of that linac, and that's the role of the physicist. So we collect data from the machine, and then we feed it into the planning computer, and then we make sure that the planning computer is correctly calculating the dose distributions that are going to be delivered to the patient. So here's a physicist doing some commissioning work. So this is when we get a new linear accelerator. We have a water tank here. This is called a blue phantom. So we call our simulated patients, we call them phantoms. And we put in measuring devices. This is an ion chamber. This is made of carbon, and inside the carbon is an air cavity. So the radiation comes in, it ionises the air in the cavity. There's a central aluminium electrode, and that measures the charge that is generated in the air cavity, and from that we can work out what dose of radiation is being delivered at the point where the chamber is being held. So if we put the ion chamber here, and then we drive it down as the radiation is coming out, we get a curve which is called a depth dose curve. So if we look at 6 MV, so we're talking about 6 megavolts of radiation that's coming out, we can see that this is the depth in our phantom, or our depth in the tissue. So on the surface, we get about 50% of the maximum amount of radiation that comes out. Here's 100%, so at 1.5 centimetres depth in the patient, we get a high amount of radiation, and then it drops off with depth. And the higher the radiation that comes out, the higher the energy that comes out, then the greater the depth of penetration of the radiation. And this is the sort of data that we use in doing our calculations. So this is where I get to the point where I try and explain to you what's the day in the life of a medical physicist, and it's actually really difficult to explain because every day is different, but I guess we all sort of have some sort of to-do list. So we go home and we have a to-do list. This is what we plan to do tomorrow when we come into work. So the first thing we might have to do is go down to the machine. Today we're rostered on to make sure the machine is performing okay before we treat all the patients that come through. So it's an early start, so we deserve a coffee after we've done our checks, and we deserve a little look on Facebook, and we can then check our emails, and then we can... Well, last night we had to work late because we had a patient that was having a very complex treatment, so we had to measure the dose in a phantom before the patient comes along just to make sure that we're going to deliver it okay. So we've got to analyse the measurements from last night. We can go off and have some lunch, and then we can come back and check a treatment plan, and then we can have a read of a journal article. Now, we might get called off to go to brachytherapy because when we're doing a brachytherapy treatment, physicists can be involved in doing the planning, but we're always there when a treatment is delivered. So if anything goes wrong with the radioactive source coming out of the treatment unit, we're on hand to take care of everything. And then at the end of the day, we'll probably check our emails and work out what time we need to come in tomorrow. That's the plan. I can tell you it never goes to plan. We might come in and do our machine checks, but they might fail. And then what are we going to do? We've got a waiting room full of cancer patients waiting for their treatment. Your job is to work out what's gone wrong and what you're going to do about it. Are you going to cancel the treatments for today, which is not obviously a very good thing to do? Do you determine that it's safe for them to continue treating the patients, and you'll fix the problem at the end of the day? So that's your first problem-solving task, and that's quite early in the morning before you've even had your coffee. So you sort it all out, say, okay, we can treat now. You go back to your desk, and you're about to get your coffee, and you find, oh no, there's something urgent I need to attend to. You've been called from the planning office. There's a patient that's got a pacemaker. Now, if you irradiate a pacemaker with too much radiation, it's not going to work anymore, so you've now got to go do some calculations and make sure the pacemaker's not going to get irradiated. But they're really nice people in the planning office. They've often got cake, so we hang around, have a little chat, some coffee and a cake, and then we go back to our desks and we analyse that data that we measured last night. But guess what? That doesn't all work out very well, so again, we've got to work out what to do about it. And then we go off to brachytherapy, but now that's been delayed because things didn't go well. And then you get to read a journal article and you talk to some nursing staff about radiation issues, and so on and so forth. So you don't need to go through the details, but basically the long and short of it is the day never goes to plan. No 2 days are the same. You spend most of your day problem solving, and you also work with a lot of people. You walk through your day and you're always meeting and talking to different people. So what you're doing is you're applying that knowledge that you've learnt during your training to solve problems. Okay, is everybody still awake? Yeah? Good, all right. So now this is the area that, of course, I get really passionate about. What's my research area? So the things that I'm particularly interested in are prostate cancer, brachytherapy, radiobiology, magnetic resonance imaging, and machine learning. So before I tell you what my project is, I've got a big project that I'm working on at the moment. I'm going to explain to you what each of these things are. Now, we've obviously covered brachytherapy, so we don't need to go into too much detail on that. Prostate cancer. So what is prostate and where is it? Here are the kidneys. So kidneys, they filter out the waste from our body, send down the fluid into the bladder. So the bladder, of course, contains the urine. When we need to pee, this is for men only, of course, not for women, the wee comes out of the bladder, it passes straight through the prostate and out to the outside world. And as I mentioned before, we've got a rectum that sits behind. So it's in a very inconvenient position in terms of getting the radiation beams in there. So what does the prostate do? Yuck, it produces liquids that goes in the semen. It also got muscles in there that helps with the ejaculation. If you don't know what that is, go look it up. Okay, so why is this a problem? Because more than 3,000 men die of prostate cancer in Australia every year. 1 in 5 men will develop prostate cancer by the age of 85. So there's a lot of prostate cancer out there. It does occur mostly in older men, so most of our patients are over the age of 60. But it can occur in younger men, particularly if there's a family history. So there are a number of treatment options. So the patient can have the prostate cut out if we're sure that the cancer is confined to the prostate and not already spread to other parts of the body. We can give external beam radiotherapy or we can give brachytherapy. So I've already explained about seeds going in. This is high dose rate brachytherapy. We put needles in. So yeah, the needles are hanging out the bottom, and this radioactive source drives under computer control and steps through the prostate one by one. The important thing to understand is we treat the whole prostate. So remember, you've got that urethra going through the middle, you've got the rectum, you've got the bladder. It's a difficult organ to treat, and given that we're treating the whole prostate, we do get side effects from treatments. These are the sort of side effects that we can get from the treatment: incontinence, impotence, rectal bleeding, needing to pee a lot at night, and so on. It does depend on which treatment the patient has, and also patient-related factors. But because of all of these side effects, we don't recommend all patients have treatment. And so it's a little bit difficult sometimes. You can say to a patient, we think that your prostate cancer is not developing rapidly. We think that it's not going to kill you. If we treat you, we will give you some side effects, so maybe it's best that we don't offer you treatment for the moment. Obviously, we'll continue to watch that patient for a while, but at some point there is that risk, of course, that they may find that their disease is progressing and we may have missed the boat for an early treatment option. So it's a very difficult decision to make for patients to decide whether to have treatment or not. So the core of our research project is about finding a treatment that cures the cancer, has few side effects, and is cost effective. So we've covered prostate cancer, we've done brachytherapy. We'll now look at magnetic resonance imaging. So this part of the screen is showing us what a prostate looks on CT. It's difficult to discriminate the prostate itself, which is outlined in red with the surrounding tissue, but on the other side is MRI. And you can actually see the prostate very clearly on MRI, and in fact you can even see detail within the prostate using MR. So MR is a great imaging modality. If we're going to treat it, we need to be able to see it. What does an MR scanner look like? So this is an MR scanner. This patient's about to have a scan of their head. They've got a coil over their head, and the MR scanner produces a magnetic field. So MR uses a magnetic field and pulses of radio waves energy to make pictures of organs and structures inside the body. And so this is a picture of a patient with a brain tumour. You can see very clearly inside the brain. Sometimes when we scan patients, we don't see very much. So the title of my talk had Madame Curie in there, it had Schrödinger in there, and I had all great plans of explaining to you how MRI works and the link between Schrödinger's equation, quantum mechanics, producing these images. But when I thought about it, I realised that we actually teach MR physics, how an MR works, in 8 3-hour lectures on our master's course, so we don't really have time to go into details today, so we'll skip over that. Okay, so that's MR. Radiobiology. What's radiobiology? So radiobiology is about the way cells respond to radiation. So if we take a tumour and we want to know if we give this amount of radiation, what's going to happen? And the likelihood of that tumour cell being destroyed depends on a number of factors. In fact, 5 factors which are in that slide. I'm not going to go through them in detail. We'll have a look at them in a moment. Usually, we don't know a great deal of detail about the individual patient's tumour. So we might know 1 or 2 things in those 5 circles, but usually we don't know everything about the tumour. So what we do is we consider all tumours the same. So we consider all prostate tumours the same, all breast tumours the same, and so on and so forth. And so we basically talk about population statistics. So we assume all tumours will behave the same way. And so when we decide on a patient treatment, we will consider only their PSA. This is prostate cancer patients. PSA is a blood test, prostate-specific antigen. The more PSA, the more cancer there is. We will consider the number and size of tumours. We can work that out by doing a digital rectal examination. Put the finger in the rectum and you can feel how big the tumour is and whether it's all over the prostate. And we can work out tumour aggressiveness. And by that we mean we take a little tiny sample of tissue from the prostate and we look under a microscope and we look at the cells to work out whether it's a rapidly, or we think it's a rapidly dividing tumour or not. And that's a little tiny sample of tissue. We usually do about 8 little samples, and of course we might miss a really aggressive one, so it certainly isn't going to tell us all the 5 bits of information that we really need to know for the individual patient. So if we did know the characteristics of the tumour for the individual patient, we can do some calculations. This is called a tumour control probability equation. We won't go into all the maths, but what I will tell you is D stands for dose, or dose rate, and if we knew something about the tumour, we could put all the values into this equation, and for a given dose, we could calculate the tumour control probability. So if we knew all of this about the tumour, and we knew how much dose we were going to deliver, we could work out what the tumour control probability was going to be. If it wasn't enough, then we would put more dose in. Conversely, if it was too much, we could not deliver as much radiation. So this is where I try and explain this concept here. So if we were delivering a small amount of radiation, then the probability of tumour control will be really, really low. If we increase the dose, then at some point we reach a threshold value or a plateau value where the probability of curing the cancer is really, really high. Conversely, if we look at normal tissues, as the radiation dose increases, then again the complication risk increases. So this is normal tissue complication probability. the gap between these two curves is called the therapeutic ratio. So our goal as physicists is to try and maximise the gap between the tumour control probability and the likelihood of complications. But our project is more involved in finding where this plateau region is. The idea is if we knew what dose we needed to deliver to reach this plateau, then that's the dose that we would deliver, because increasing the dose any more would just increase the probability of complications. Okay, so that's a snapshot of radiobiology. It's quite a complex field, but anyway, that gives you a little bit of a feel. So now we're going to have a look at what machine learning technique is. So again, we've got another video. This is about skin cancer.
We built an algorithm that, using a single image, can classify skin lesions as cancerous or benign as accurately as board-certified dermatologists. Neural networks, also known as deep learning algorithms, are a type of very powerful machine learning algorithm. And the way these work is that they are fed massive amounts of data, and they are told what they are looking at. The hope is that this would be a starting point into bringing AI and computer vision to healthcare and really extend the reach of providers well outside of the clinic.
Pretty cool, hey? We come across machine learning in our days all the time, even things like traffic lights and all sorts of different applications of machine learning. It's probably the next big thing of the future, and we're applying it in the health field quite regularly these days for predicting all sorts of things, in predicting which types of treatments are best for patients. But it's still at the infancy in the health field, and I think we tend to be a little conservative in the health field because we really need to be sure about what we're doing. I mean, if the traffic lights fail, it's a bit of a nuisance, but hopefully nobody dies. But in our field, people can die, so we have to be really sure and really confident about what we're doing. Now, in this particular video, they showed the development of an algorithm. We call it a predictive model. And what the model was built, the algorithm was built by taking pictures of spots, and we knew whether the spot was a cancer or not a cancer because we'd done a biopsy. So this is called the ground truth. So these patients actually had biopsies. But in the future, the idea is to use the algorithm to predict when a spot is a cancer or not a cancer. If it predicts it is a cancer, then of course the patient would have a biopsy, confirm that it really is a cancer, and then they would have the appropriate treatment. So, okay, so I've told you a little bit about each of these things, so now onto my project. So going back to prostate cancer. So cancer in the prostate is not actually what kills the patient. The cancers can leak outside the prostate and they can spread to other parts of the body. So usually they first go into the bone and into lymph nodes, but then they can spread to the liver and brain and lungs. And it's actually those metastases that actually kill the patient. So what we're trying to do is we're trying to destroy the cancer before it gets outside the capsule and then eventually kills the patient. So you might say, well, if we know where the tumour is, in this case the tumour is there, then we could potentially just zap that tumour instead of the whole prostate. Oops. Oh, yeah, that's right. That's what I was going to say. So, yes, from our MRI, we can actually see where the tumour is. So why not just zap the tumour instead of the whole prostate? But one of the problems is the fact that prostate cancers do tend to have lots of little tumours also in other parts of the prostate. So in our project, we're proposing that we will still zap the tumour itself with a very high dose of radiation, but we'll deliver a smaller dose of radiation to the rest of the prostate. So basically, the larger the prostate, the more radiation the larger the tumour, the more radiation you need to put in there to destroy the cells. So we can get away with smaller doses of radiation for those little tiny tumour deposits. So we could use MR, that's great, But here's another cool video. Can we trust our eyes? We're looking at those grainy grey images. Do we believe what we're seeing? Very interesting, eh? So I think the answer is no. So MR images can have artifacts in them for all sorts of reasons, and so just looking at the grayscale images is not providing us with that high-level accuracy that we need for the way we practice radiotherapy. So what we're interested in doing is developing predictive models using machine learning techniques. So they analyse these MR images and find the tumour. So again, we have to have our ground truth information. Now, this is actually a piece of tissue that has been taken, or it's actually a prostate that we've taken out of a patient and chopped it up. Now, obviously, if the patient's had their prostate removed, they're not going to have radiotherapy. But remember, to build the predictive model, we have to have ground truth so that future patients that are having radiotherapy, we know where their tumour is. So we want to predict the tumour on MR. We're using the histology, which is the prostate, for our ground truth to develop our predictive model so that in future patients we just do the MR and we can work out where the tumour is. Okay, so that tells us where the tumour is, so we can zap it with this high dose of radiation. But now we want to know what dose of radiation. So again, going back to what I was saying before, we could use population statistics, but how about we now tailor the radiation dose to the specific tumour? So let's find that plateau on the probability curve so that that patient gets the amount of radiation that they need for their tumour. So this is going back to our radiobiology equation, and so this is going to tell us what dose that we need to deliver. Which is great, but what we need to do is we need to determine the biology of the tumour. So those little tiny bits of tissue, the biopsy, is not enough. So, so just remember we're trying to find this, find this peak plateau region on the curve. So going back to our predictive model, so before I was saying we're going to take MR data, we're going to use the predictive model to find the tumour location. We're now actually, rather than just using MR, we're going to use a thing called multiparametric MRI. I'll explain what that is in a moment, plus the histology, to not just deliver to find the tumour location, but also determine something about the tumour biology. This is multiparametric MRI. Basically, it means that we can do lots of different sequences. So the patient is actually in the scanner for about 30 to 40 minutes, and we do different sequences, and they tell us little different bits about the actual way that the prostate is structured, and from that we can deduce the biology, or at least that's the plan. On the right-hand side of the screen, I've actually put some PET images. PET stands for positron emission tomography, which is nuclear medicine, but I'm not going to talk about that in this presentation. So bringing it all together, we have all of the MR images from all of the different sequences here. We apply image processing techniques. Machine learning requires feature extractions. We look at features in those, and we combine that with our histology, our ground truth information, to develop our predictive model to determine the information that we need for the radiobiology calculation. And this is called radiomics. It's called quantitative MR, so we get numbers rather than just looking at the grey images to give us some information about the biology of the disease. So when we've been taking patients that have had their prostates removed and chopping them up, the pathologist marks it up for us. So the pathologist marks up where the tumour is, and they tell us something about the structure of the cells. So this is our ground truth histology. And we take the magnetic resonance imaging, all the different sequences, and again we can produce what we call pharmacokinetic maps. These give us numbers now, so instead of those grayscale images, we can analyse the data from these images. And we divide the images up into blocks, which are called voxels. So what we have to do is match each voxel from each of the images, and we put them up into a matrix. So the red represents our MRI. So voxel number 1, let's say this is voxel number 1. This is from a T2 weighted image. That's a signal intensity. And again, I won't go into the details of what all of these numbers mean, but they just simply come from all the different imaging sequences that we've done. And then we apply our machine learning techniques using the ground truth histology. So we know whether this particular voxel contains tumour or benign tissue, what its tumour cell density is. Gleason's score tells us something about the aggressiveness, and hypoxia is about whether it's got oxygen in it or not, and I'll explain what that is in a moment. So essentially we get all the voxels together and we build a 3D map, and so we can compare each voxel, all the different numbers, and you apply a machine learning technique to predict in future patients what those biological parameters are. It's a very complicated process bringing all of this data together. The histology, of course, looks absolutely nothing like the in vivo. This is the MR that they have before they have their prostates removed. So we go through a complex process. So you imagine you take a prostate out of the patient, it's all squishy, and fluids leak out of it, and it gets deformed. So we have to apply morphology, lots of different algorithms to match it all together to be able to develop our machine learning algorithms. So this is some work that one of my PhD students has been doing. This is looking at 4 different machine learning techniques to predict tumour cell density. So we've actually taken the histopathology, we've applied some machine learning techniques. So we've counted the number of nuclei in the cells, basically. That tells us our tumour cell density. And this is the model performance. Similarly, Gleason grade is about how aggressive a cancer is. So we look under the microscope and we see the cells are evenly packed, which means it's unlikely to be an aggressive tumour. If the cells are irregular and loosely packed, then it's more likely to be a more aggressive disease. Again, applying machine learning techniques, we've been able to do this. We can predict where the tumour is and the grade of the cancer as well. I thought I'd just throw in hypoxia because this brings in another element of our work. So hypoxia basically means that a cell is devoid of oxygen. When cells are devoid of oxygen, it means they're more radio-resistant. And what that means is you need to put more radiation in to destroy the cell than a cell that is devoid of oxygen. So a hypoxic cell has not got oxygen in it. Now, unfortunately, when we take a prostate out of a patient, it's now essentially dead. And you can't really work out whether that tissue has got oxygen in it or not before you took it out of the patient. So this is our MR scans that we can use to identify hypoxia, but we need the ground truth information. So we've been dabbling a little bit with genetics. And what we found is that, and this is from the literature, this is not actually our work, but what we found is that certain genes are expressed when there is a lacking of oxygen. So these genes are suggesting that these cells were devoid of oxygen. So in other words, they're hypoxic. So this then, if we take the samples out of our prostate, becomes our ground truth. And this is what we've been doing here. We take what we call punch biopsies. These are holes there, and we send them off to the labs, and then they give us the genetic signatures, and we can work out the ground truth. Was that cell devoid of oxygen or not? Okay, so this is what it all looks like, or what we hope it's going to look like at the end of the day. So this is our vision for the future. This is called the BERT project. We all have to have acronyms. People remember your project if it's got an acronym. So I hope you all go away and go, Annette talked about the BERT project. Okay, so BERT, it stands for biofocused radiotherapy. Bio meaning the biological part of it. Focus means we're focusing on the tumour, and radiotherapy, of course, we're delivering radiation. So in future, the patient will come in, they will have all those MR scans, that's the quantitative imaging. They will then have a treatment plan generated. Now, we like brachytherapy, but equally you could use external beam. Now, we've got all this imaging that we did before the patient had treatment, so what we can do is we can then image them at several points after they've had the treatment to see if there's changes in those parameters in the MR imaging to work out whether the treatment worked or not. So this is getting to the end now. What do I love about my job? You probably gathered I love working with different people. I love working with lots of people. These are people who have been involved in my research, or some of the people who have been involved in my research project over the years. So I won't go through them all, but I'll just mention a few people. So Hayley up here is my postdoc. She's actually a bioengineer from New Zealand. Scott is our radiation oncologist. He's down in Melbourne. Darren is our statistician. He's in Queensland. And Yu Sun is our star PhD student who's been doing all the machine learning techniques. Gary is an MR physicist in Liverpool. We've got some more engineers here. These are computer scientists who are in Vienna. These are some mathematicians at Monash University in Victoria. Declan is our champion surgeon down in Victoria. Mary is another MR physicist. She's in London. Martin is a physicist and a radiobiology specialist. He was my PhD supervisor. And Emily is another one of our PhD students also in Perth. And there's lots of other people like our pathologist and our radiologist that I didn't have photos for. But what's really important is the patient. So we recently analysed the data from from a whole bunch of patients that we treated in the early days of our prostate program. So remember I told you I was the first physicist involved in the modern techniques of prostate brachytherapy where we implanted those radioactive sources. I told you at the beginning of my presentation that we used graph paper, we had hand calculators, it was all very crude in those days, and that's what we did, but we treated a whole bunch of patients. So we followed those patients very closely to see how they did, and we published this paper last year. And for those patients that we had data on, that we'd followed them for more than 10 years, despite the crudity of the way we did things, 96% of them, of those with low-risk prostate cancer, are free of prostate cancer today. How cool is that? Okay. So, as I said, this is the end of my presentation now, so I've only touched the tip of the iceberg today. There's a huge amount more under the water there, which obviously I can't cover, but I'll be very happy to talk to you about anything else. Are we cool? This is the picture I showed in the beginning. This is probably what I thought a medical physicist looked like before I went into medical physics. We actually look like this. So these are some cool dudes from our National Dosimetry Service. When we're not measuring Linux, we do all sorts of cool stuff together, which is really a lot of fun. So again, I have to acknowledge the many people that have been involved in my work over the years and the funding partners. We have an NHMRC grant to continue our work, and also PhD students that love getting distracted from writing up their theses. Yusan and Yessa helped me put together this presentation. Thank you very much. That's it.